Hi there, my name is Dr. Simon Dingsdag. I'm part of the Studies in Translational Microbiology and Emerging Diseases Research Group, or MICROS, at the University del Rosario, along with Dr. Oliver Clay and Dr. Gustavo Quintero. Today I would like to introduce our ongoing research project, which is a multidisciplinary effort of surgeons, undergraduates, a microbiologist, and a computational biologist from the schools of Medicine and Health Sciences, as well as the Faculty of Natural Sciences. The project consists of two broad parts, a microbiome metabolome component and also a human genetics component. Firstly, I will briefly introduce the microbiology of the biliary tree. Later in the presentation, Dr. Clay, our computational biologist, will continue to describe more specific analyses of how to map gallstone diseases to the underlying human genetics through haplotype analyses. Right, so the biliary tree consists of the liver, which produces the bile, the gallbladder, which stores the bile, and the ducts in between. In particular, we are interested in conditions resulting from the formation of stones in the biliary tree, which form through precipitation of various metabolites, including cholesterol or bilirubin from the bile. Since bacteria are present in varying numbers in the bile and also inside gallstones, we hypothesize they may be involved in the pathology of gallstone formation or downstream morbidities. It is known that the microbiome is complex and actually surprisingly has been quite varied in published studies. So, in order to clarify, we wish to create a more detailed map by characterizing the microbiome of a Colombian cohort of patients. We're going to include bacteria from their feces and also their dental plaque, and finally we will overlay the metabolome of the bile and gallstones with the final aim to determine, in essence, if bacteria have a role in gallstone formation and related downstream pathologies. As noted, there are a variety of different types of gallstones, broadly characterized by the ratios of the components they are made of. For example, cholesterol gallstones have a high amount of cholesterol in relation to other metabolites. So in general, the relationship between underlying genotype, environmental factors including diet, as well as the microbiome are complex. However, the prevailing model is summarized by the triangle on the right-hand side, which contains three axes that represent the concentration of cholesterol or other groups of metabolites. In essence, in healthy bile, these metabolites are soluble, so that during the shift to dysbiosis, even small increases in particular metabolites, for example cholesterol in proportion to total bile acids and lecithins, leads to precipitation. As this precipitation process continues, gallstones may form from these precipitates or other non-soluble metabolites. As mentioned, bacterial presence, particularly in the bile, might result in consumption or even production of metabolites that contribute and even accelerate gallstone formation.
Hello, and thank you for your interest. I will be talking about the genetic aspect of this project, and especially the part that we can already do now before the results from bacteria from the microbiome come in, and using public data, which is really looking at differences at the genetic level that could correspond to differences in gallstone risk or prevalence. There are now quite a number of studies accumulating, showing results of this kind. And one analysis we are doing is looking at genes that have been already highlighted as a result of genotype-phenotype studies, or is at the cellular or molecular level, focusing on functional roles of the gene's protein products. The map you see here shows population-level phenotypic information in the form of percentages of prevalence for a gallstone formation or discomfort. While the colored areas, the colored countries or regions, show where there has been the collecting genotypic data in the local populations, with sample sizes that allow one to really see with some kind of confidence which haplotype frequencies exist and how they differ among the different regions. This is the 1,000 Genomes Project, in which just over 5,000 chromosome sets were sequenced completely, and the differences stored in a public database, which we refer to here as 1kg. Of note, there is a marked tendency for populations with a component of American ancestry, and particularly women, to have distinctly higher gallstone risk. As you have seen in the previous slide, the gallstone disease is not a particularly rare disease. Correspondingly, one is interested in a common variation, in other words, SNPs in which the minor allele frequencies will be greater than, for example, 3% or 5%. This means that with relatively small samples of individual populations, one can already get a good idea of what these frequencies will be. The analysis is actually simpler if one just takes those single nucleotide polymorphisms, or SNPs, that are biallelic. So they have only two states, either 0 or 1 if one wants to look at it numerically. And in this case, a very simple plot, the minor allele frequency plot, which shows the frequency at each position along the chromosome that one is interested in, is seen to create a landscape of blocks or plateaus that can be quite different from one world population to another. Such a block-like or mosaic structure was predicted already in the 1980s, but was not actually shown until population sequencing could begin of whole genomes or whole chromosomes, so around 2000. And in 2001, there were a number of different papers, some of them from Eric Lander's group, that clearly showed the haplotype block mosaicism of human chromosomes. This means that there is also often a very strong correlation or linkage disequilibrium among SNPs within the same block. And these blocks can often be very long. And as the case in the Gefferin locus, for example, they can also have only two states, which means that you have most of the common SNPs having either all the major allele frequency or all having the minor allele frequency. To some extent, this obviously simplifies the analysis and simplifies the vision. The analysis also becomes simpler if one can just abstract from the actual nucleotides A, C, G, and T and just look at each position where there is a common polymorphism and just see which one of them is the minor or the major allele. For example, we give the number 0 to the major allele and 1 to the minor allele. Then the extreme case is when one haplotype or long segment of successive SNPs is all 0, 0, 0, 0, and the other one is all 1, 1, 1, 1. That is the simplest limiting scenario. In reality, there is some variation, as you can see in the plot on the right side. And there you can see that the intra-class variation in minor allele frequencies is much less than the inter-class variation. So that is the idea. In brief, we looked at 15 characterized human protein coding genes that had some kind of a association or functional role in processes that are necessary for gallstone formation, or that could prevent gallstone formation. Some of them are shown in the diagram here, where they are mapped onto the enterohepatic circuit. This slide has a video embedded, which may work depending on the way your computer is set up, and that shows the chromosomal segments that contain each of the 15 genes that we looked at in this first instance. Even if you cannot see all of them, this first plot, or rather 5 plot set, for corresponding to different superpopulations and showing 1kg data, shows the kinds of landscapes that you can find and that also characterize many of the other genes in this video. That is, not just the genes, but often long regions around them, or containing part of them and containing partly intergenic DNA. On the vertical axis, you see the minor allele frequency in each of the 5 superpopulations, and at the bottom you see a plot from UCSC Genome Browser that shows where the genes are. Although it would go beyond the scope of this presentation to go into more details, there is a clear advantage of using haplotype analyses over typical SNP by SNP analyses, because one gets a very good idea of the structure, and also one can see possible strategies involving different SNPs that have to be conserved together in order for the strategy to work. And again, very often one can find regions in which there are essentially only 2 or 3 different haplotype variants over a relatively long stretch encompassing a gene, for example. And these last 2 slides show some details of the project within which this work was done. We would like to thank the Universidad de Rosario and also the Metabolomics Core at the Universidad de los Andes and the Medellín Hospital. And thank you also for listening.